We've talked a bit about ATP and how it's necessary to do work in the body. So let's talk about how ATP is actually produced or synthesized. To be clear, there's two ways to synthesize ATP. One of them is via oxidative phosphorylation, which happens in the mitochondria, and then substrate level phosphorylation, which tends to happen within the cytoplasm of cells. That is to say, the interior of a cell bound within the plasma membrane, but certainly not within an organelle and not within the organelle that we know as the mitochondria. Cellular respiration incorporates three phases: glycolysis, the Krebs cycle, and the third one is the electron transport chain and oxidative phosphorylation. The electron transport chain and oxidative phosphorylation are both part of the third phase. So we have glycolysis, the Krebs cycle, and then the electron transport chain / oxidative phosphorylation. And while oxidative phosphorylation, which we see right here in this image, this is an image of the mitochondria, while this is the last phase of cellular respiration, I prefer to start the discussion here because it really informs us what's going on in the two previous phases, glycolysis and the Krebs cycle. So once again, oxidative phosphorylation occurs in the mitochondria. There are a litany of mitochondria within individual cells in our body. There are certainly some cells like red blood cells that do not have mitochondria, but most cells do have mitochondria. The anatomy of the mitochondria is important to understand for the nomenclature I'm going to use in this discussion. So here is an image of a mitochondria. This is the outer membrane of the mitochondria, as we see right here. And this right here is the inner membrane of the mitochondria. So we have an outer membrane and an inner membrane. Interior, or within the inner membrane, we have the mitochondrial matrix. So all the space in here that is surrounded by the inner membrane is the matrix. Between the inner membrane and the outer membrane is the intermembrane space. Now, one thing we can see already within this image is that there is a super high concentration of the hydrogen ion within the intermembrane space. Keep in mind, inter means between, so intermembrane is between the two membranes. There's a super high concentration of hydrogen ions or protons within the intermembrane space, and a much lower concentration of hydrogen ions within the matrix. Additionally, we have a number of proteins right here that I have labeled 1, 2, 3, and 4. These are complexes known as complex 1, complex 2, complex 3, and complex 4. And these are what make up the electron transport chain. This right here in purple is ATP synthase. Once again, because it has the suffix ASE, that's indicating to us this is an enzyme. This should not be confused with ATPase. We've talked about the sodium-potassium ATPase, the calcium ATPase, the hydrogen-potassium ATPase, and the potassium ATPase in the beta cells of the pancreas. And there's a number of others. Those ATPase is an enzyme that breaks down ATP. ATP synthase builds or produces or synthesizes ATP. And I am working backwards. Once again, I'm working backwards by talking about oxidative phosphorylation and the electron transport chain prior to the other phases in glycolysis, but I'm also going to slightly work backwards with regards to what is happening within the mitochondria. That is to say, specifically, oxidative phosphorylation. One very important thing to point out is energy is required to produce more energy. So the goal of ATP synthase is to produce chemical energy in the form of ATP. So what are we going to use to produce that ATP? Well, what ATP synthase uses is kinetic energy, and specifically the kinetic energy of the protons that are in the intermembrane space, otherwise known as hydrogen ions, the kinetic energy of these protons moving passively from an area of high concentration into the matrix where there's a lower concentration of hydrogen ions. Because the proton is charged, it requires a channel, and the channel it uses to move from the intermembrane space into the matrix is ATP synthase. So, as the hydrogen ions move passively via ATP synthase into the matrix, they are creating kinetic energy. And that kinetic energy is going to be harnessed to couple one phosphate to ADP to create ATP. And that's what we see right here, one inorganic phosphate and one ADP, otherwise known as adenosine diphosphate. If you couple or attach this inorganic phosphate to ADP, we're going to make ATP. But we need energy to do that. The energy that is going to be used to do that is the kinetic energy of the hydrogen ions moving down their gradients into the matrix. On the face of it, that may not seem too complicated because we've talked about kinetic energy being used to push ions or molecules against their gradients or to help facilitate some other metabolic processes. But how do we maintain this high concentration of the hydrogen ions out here? And this is very similar to the sodium-potassium pump and the gradients of sodium and potassium with regards to many cells in the body. Once again, sodium moves passively into cells, potassium moves passively out of cells. And if this continues to happen, the cells would lose their chemical gradients. So to maintain those chemical gradients, our cells have the sodium-potassium pumps, which pump sodium against their gradients and potassium against its gradient. So let's just focus on sodium for this example. It keeps pushing sodium out of cells, to ensure there's a higher concentration of sodium outside of cells than inside of cells. And that's exactly what the electron transport chain is doing. The electron transport chain pumps hydrogen ions from the matrix into the intermembrane space. The electron transport chain is responsible for moving hydrogen ions against their chemical gradient into the intermembrane space. But like the sodium-potassium pump, we need energy to move things against their gradients. And the energy that the electron transport chain is going to utilize is the energy imparted by electrons being dropped off from one complex to the other. And this is where we're going to get to these two molecules, NADH and FADH2. NADH is nicotinamide adenine dinucleotide. FADH is flavin adenine dinucleotide. These are shuttling electrons from glycolysis and the Krebs cycle. So to be clear, once again, glycolysis and the Krebs cycle are the first two phases of cellular respiration. And one of the main themes of those processes is to liberate electrons that are grabbed by NADH and FADH2 and delivered to the electron transport chain to power the movement of hydrogen ions into the intermembrane space. So if we just look at NADH for a second here, as NADH comes to the electron transport chain, it drops off an electron that moves to the first complex. That electron is going to then bounce from one complex to the other until it arrives at oxygen. Oxygen is the final electron acceptor in this process. And it is oxygen that is pulling those electrons down this electron transport chain. And as the electrons move from one complex to the other, energy is released. So we have this electron being dropped off by NADH to this first complex. And then that electron is going to move to the next complex. And as we can see, that electron bounces or gets passed onto the next protein in this series. This is the electron transport chain. Every time that electron is liberated from one of the complexes, energy is released. And that energy is going to be used to move or push hydrogen ions uphill or against their gradients. That looks something like this. Now, to be clear, hydrogen ions are moved into the intermembrane space via more than just two of these complexes, but for the sake of space and being able to really see what's going on here, I've only drawn two arrows here. But to be clear, the main goal of the electron transport chain is using the high energy derived from these electrons to push hydrogen ions into the intermembrane space. Now, we have talked about secondary active transport. I'll put a link up here for that video that if you want to review primary and secondary active transport. Kinetic energy is valuable in moving things against their gradients, especially in the absence of chemical energy, or if we want to save that chemical energy for other things. So kinetic energy, like for example, the sodium glucose transporter that we talked about, is only going to be functioning if we first have primary active transport. That is to say, the primary active transport maintaining sodium's high concentration out of cells. And that's the same thing here. This kinetic energy created by hydrogen ions moving down their gradients is only possible if we're first if the cell is continually pumping hydrogen ions into the intermembrane space to maintain this high concentration of hydrogen ions. So the energy here in the electron transport chain is the primary active transport, whereas this is really secondary active transport. Now that we have that, let's take a look at the first two phases of cellular respiration, and we'll start with glycolysis. So with regards to glycolysis, and really with regards to cellular respiration, any of the phases, glycolysis, the Krebs cycle, or oxidative phosphorylation, one thing we want to always have the forefront of our consciousness is the equation for cellular respiration. C6H12O6 plus 6 oxygen creates 6 carbon dioxide, 6 water, and ATP, and certainly heat. And I realize I don't have heat drawn in here, but please understand that during this process, heat is produced. This equation is not specific to glycolysis, which we see right here, and this is going to be a very abbreviated description of what occurs during glycolysis. But during glycolysis, we have substrate level phosphorylation. Within the mitochondria, we have oxidative phosphorylation. When I say within the mitochondria, I'm saying specifically via ATP synthase, because what we'll see with the Krebs cycle in a moment, the Krebs cycle also occurs in the mitochondria, though it occurs exclusively in the mitochondrial matrix, whereas glycolysis occurs in the cytoplasm of the cell. I did mention that red blood cells lack mitochondria, and the red blood cells, otherwise known as erythrocytes, are going to produce ATP glycolytically. That is, say, within the cytoplasm, because they don't have mitochondria. This bubble right here has nothing to do with glycolysis. I'm just trying to demonstrate what substrate level phosphorylation is. That is to say, it's taking a phosphate from one substrate or one high energy molecule, in this case creatine. This is known as creatine phosphate or phosphocreatine. And it's transferring that phosphate to ADP, creating ATP. And certainly the substrate without the phosphate, which is just creatine. So this is an example of substrate level phosphorylation, and the ATP that are produced during glycolysis are produced via substrate level phosphorylation. Now, I want to be clear, glycolysis can occur with other molecules, it can occur with lipids and proteins, but oftentimes it's using glucose. And as far as glucose goes, we've talked about monosaccharides, glucose, galactose, and fructose, which are all very similar. They're all C6H12O6, but slightly different. 80% of the monosaccharides circulating through the blood are glucose molecules. When we absorb fructose and galactose via the digestive tract, they immediately go to the liver. All of the galactose is converted to glucose in the liver, and much of the fructose is as well. So we're generally using glucose in glycolysis. And glucose carries a ton of energy, 686 kilocalories per mole of glucose. And if we were to combust that or oxidize that in one fell swoop, there would be a ton of energy release, but our body could not use it because it can't capture, hold on to, or store that energy within ATP. And that's really the role of ATP, is to store that energy in smaller components in usable forms. There's many steps in glycolysis, but the end products of glycolysis are 2 pyruvate molecules, 2 net ATP molecules, and 2 NADH molecules. What I mean by 2 net, there are 4 ATP produced during the litany of enzymatic steps in glycolysis, but in the process, the cell is using 2 of those ATP. So 4 produced, 2 are used, so we have 2 left over, and that's why we refer to it as 2 net ATP. Keep in mind that glucose is a 6-carbon molecule, and at the end of glycolysis, we are left with 2 3-carbon molecules, and those 3-carbon molecules are pyruvate. So we have 2 pyruvate molecules, 2 net ATP molecules, and 2 NADH molecules. So one thing I neglected to say when we talked about oxidative phosphorylation is going to create upwards of 34 ATP molecules. And as we see here, glycolysis is only producing 2 net ATP molecules, which doesn't sound very valuable, which, truth be known, it's not very valuable when we can compare it to the oxidative phosphorylation in the mitochondria. But these NADH molecules, these 2 NADH molecules that are created, are very valuable because they are transferring those electrons to the electron transport chain, which are going to be utilized to continually pump hydrogen ions into the intermembrane space. Another thing to keep in mind is that glycolysis does not require oxygen. We certainly saw oxygen is used within the electron transport chain, and that's certainly directly related to oxidative phosphorylation. We need oxygen within the mitochondria to pull those electrons downhill within the electron transport chain, or with respect to the electron transport chain. And that's why we breathe in oxygen. That is the only reason we breathe in oxygen, to create that ATP within the mitochondria. Glycolysis occurs in the presence or absence of oxygen. In the absence of oxygen, pyruvate enters what's known as lactic acid fermentation, and I'll do another video on that at a later time. In the presence of oxygen, pyruvate is going to proceed towards the Krebs cycle. Once again, in the presence of oxygen, pyruvate is going to proceed towards the Krebs cycle. The Krebs cycle occurs within the mitochondrial matrix. It's also known as the citric acid cycle, and also known as the tricarboxylic acid cycle, which is hard for me to say. So I generally say just Krebs cycle. Krebs cycle is easier for me to say than even the citric acid cycle. So from here forward, we'll call it the Krebs cycle. Pyruvate does not directly enter the Krebs cycle. What enters the Krebs cycle is what's known as an acyl unit. This is a 2-carbon molecule that enters the Krebs cycle. Upon entering the Krebs cycle, it is going to react with a molecule known as oxaloacetate. Oxaloacetate reacting with this acyl unit is going to create citric acid, otherwise known as citrate. Once again, oxaloacetate plus this acyl unit, which is entering the Krebs cycle, creates citrate. This is a cycle, and it's going to proceed along this cycle. The end product of this cycle is oxaloacetate. Oxaloacetate is both the end product of the Krebs cycle and one of the initial reactants, because it's going to react with this acyl unit. The Krebs cycle is a multifaceted enzymatic process that we are not going to go into. It's not necessary for our understanding of ATP synthesis with regards to human physiology. I will provide more detail on this in a subsequent video, but it's going to end up creating one ATP molecule. Once again, like glycolysis, that's not very many ATP molecules. So the goal really, what we're trying to achieve within the Krebs cycle is not the ATP. I'm not saying it's totally meaningless, but it's these NADH molecules and these FADH2 molecules. The Krebs cycle produces 3 NADH molecules and 1 FADH2 molecule. Once again, these molecules are transporting electrons to the electron transport chain, and that is uber important if we want to keep or maintain that hydrogen ion concentration within the intermembrane space of the mitochondria. So the Krebs cycle produces 3 NADH molecules, 1 FADH2 molecule, 2 carbon dioxide molecules, and this 1 ATP. But where does this acyl unit come from? Well, in the presence of oxygen, pyruvate is going to react with coenzyme A to produce acetocoenzyme A. This is a 3-carbon molecule. Pyruvate's a 3-carbon molecule. This process is also producing NADH and CO2. So once again, pyruvate in the presence of oxygen will react with coenzyme A to produce acetocoenzyme A, an NADH molecule, and 1 carbon dioxide molecule. The acetocoenzyme A is going to be enzymatically broken down into this 2-carbon acyl unit, which will then enter the Krebs cycle and react with oxaloacetate. So how many ATP molecules are created in the Krebs cycle after the breakdown of 1 glucose molecule via glycolysis? If you said 2, you're correct, because at the end of glycolysis, we are left with 2 pyruvate. So everything I've been describing here happens twice, because at the end of glycolysis, we are left with 2 pyruvate molecules. So if we have 2 pyruvate molecules, we're going to end up with 2 acetocoenzyme A molecules. Each of those are going to be converted to the 2-carbon acyl unit. They will individually enter the Krebs cycle. So one enters the Krebs cycle, creates these 3 NADH, 1 FADH2, 1 carbon dioxide, and 1 ATP. The other one then enters, that is to say the other acyl unit enters, and we produce 3 more NADH, 1 more FADH2, 1 more CO2, and 1 more ATP. So after the breakdown of glucose via glycolysis, we produce, or I should say, 2 pyruvate are produced, and in the presence of oxygen, proceed towards the Krebs cycle, and those 2 pyruvate molecules will ultimately create 6 NADH, because this is happening twice, 2 FADH2 molecules, 2 carbon dioxide molecules, and 2 ATP molecules. In addition to that, all of this is created twice. So the conversion of pyruvate to acetocoenzyme A is going to create 2 NADH, because we're going to have 2 pyruvate molecules and 2 CO2 molecules. So where do the 6 CO2 molecules come in the cellular respiration equation, which once again is C6H12O6 plus 6 oxygen gives us 6 CO2, 6 H2O, and ATP. We can count those CO2 molecules. There's going to be 4 via the Krebs cycle because this is happening twice. One cycle through produces 2, another cycle produces 2 more. So the Krebs cycle is producing 4 CO2 molecules. And here, the conversion of pyruvate to acetyl-coenzyme A, when it happens twice, because there's 2 pyruvate molecules from glycolysis, this produces 2 CO2. So we're going to have 4 CO2 molecules from the Krebs cycle and 2 CO2 molecules in this conversion, which is a total of 6 carbon dioxide molecules. Arguably, the single most valuable thing, or the most valuable things within this whole process, are going to be the 6 NADH molecules and the 2 FADH molecules that are created, plus these 2. So it's actually 8 NADH, 6 plus 2 is 8. And the 2 FADH2 molecules, plus those 2 NADH from glycolysis, are all moving to the electron transport chain to help push those electrons into the intermembrane space. I will put links to my gradient video. Understanding gradients with regards to the electron transport chain, or more specifically to ATP synthase, is super important.